All right, chapter 11 is microbe-host interactions. This is a time to remember that for every cell in your body that's human, you have 10 bacterial cells. And so it kind of throws into question, right? What is it to be human? Are we just a vessel to carry around bacteria? I think we're more than that, but we do have good relationships with lots of bacteria. So in this chapter, we'll get to explore a little bit more about those good relationships and then also talk about microbes as pathogens. The next two chapters will be moving into then deal with the immune system, so how our body is able to fight them off, right? So we went from cleaning surfaces to taking antibiotics. Here we're looking at our interactions, and I do think your book does a nice job of pointing out that they're not all bad. And then our next chapters are how our body tries to kill bacteria and viruses, etc. So as we jump in here, just remember, right, you as a human host for pathogens provide a variety of different niches and microhabitats that these organisms find useful, that they appreciate, right, and will move into. And a lot of times this is good, right? This is what provides our normal resident microbiota or our microbiome. So this image is from a meeting on the Human Microbiome Project. And this is kind of neat to me because when I was in college, they just came out with the Human Genome Project. It was just wrapping up. And so we've kind of moved on from just looking at our own genes now to looking at the genes and therefore the organisms, the microbes that live inside of us. So there are a lot of really good, important mutualistic or commensalistic relationships, and they can help us. And one of the big ways they help us is actually through a microbial antagonism. So they like us as a habitat, as a niche, and so they're willing to fight off other microbes to kind of save their space, save their resources. And so in some cases that really does protect us from some other bugs. So the classic example is in the vagina. We have a species of bacteria called lactobacillus, and it produces lactic acid, which lowers the pH. That low pH is going to prevent yeast from growing. And so really common way to get a yeast infection is to take an antibiotic for strep throat, something else, right? But it happens to wipe out your resident microbiota that should be living in the vagina. And instead, when that pH is allowed to rise, then yeast, a fungal infection, can move in. So these are important relationships. One of the things we will see, though, is for as many times as we get along with our microbes, there are times when if our immune system is weakened or if that microbe ends up in the wrong part of the body, that they can go from friend to foe, right? Our resident microbiota can become what we call an opportunistic pathogen. They can cause disease. Classic example, E. coli. We love having it in our large intestine. It helps us with digestion. It makes some nutrients, but it is the leading cause of urinary tract infections. So if it gets into the wrong location, it can be problematic. So I can move on. Sorry. So one of the things we're looking at, it's interesting. So even in the previous version of this textbook, we still talked about the fetus as being sterile, and then we talk a little bit about the colonization from there. It's kind of interesting. They're actually starting to think, right, amniotic fluid and the fetus may have some normal microbiota. So that may be changing some. But I like this because we'll talk about a lot of these places that we know have microbes, but now we're starting to use different technology, right? Using DNA, maybe looking for microbes that don't culture in the lab nicely. And we are starting to find them in other locations. Oh, look, there's the bladder, right? We've always said the bladder is sterile too. So some of the places that we do know for sure, absolutely, definitely have bacteria, right, along on the skin. Notice this is all up in the epidermis. These places where it goes down deeper along glands, those are just in-pocketings of epidermis. So we don't have typically microbes down in the dermis. Lining the oral cavity and the pharynx, we absolutely have a whole variety of bacteria that live in there, the large intestine as well. We were just talking about E. coli. This is an interesting one because you can actually have people who, say, carry around some strep species that they're just carriers. It doesn't make them sick, but it absolutely can transmit and make other people sick. But that's just one of the many microbes that would live here. We already mentioned the female reproductive tract, but also the male reproductive tract harbors bacteria. One of the super interesting things, I should point this out, is that actually the normal microbiota of the vagina actually changes as labor is approaching. So something about the hormone conditions of pregnancy actually alter the microbes that are there. And as we'll see here in just a minute, that might be really important in colonization of the newborn. Okay, if you're more of a graph person than the picture person, feel free. The nice thing here is they are pointing out some of the genera, that's plural for genus. So again, on your skin, we've been working with Staphylococcus epidermidis, Micrococcus luteus, right? So there's common bacteria that live on us. In the oral cavity, more Streptococcus, Lactobacillus, you might recognize. Large intestine, we should have Escherichia in here somewhere. There it is, Escherichia, E. coli, etc. So you can look through there if you're interested in what particular strains, and they also give some really interesting and common examples. Okay, so this idea of initial colonization. So again, we're kind of exploring now, maybe the fetus isn't quite sterile, but in general, there's very few, if any, microbes, right, on this baby when it's actually in the amniotic sac. So the initial colonization is considered as the baby's coming out the birth canal. And as I was just saying, the bacteria then in the birth canal are super important in kind of the bacteria that start off this baby's life. In fact, there's some studies looking at differences between cesarean section and vaginal birth and what role that might play later in allergies, asthma, etc. Some really interesting research. And some of the researchers who were doing that, I had read at least one story where his wife did have to have a C-section, and they actually did a vaginal swab and then wiped it all over the baby because they wanted the baby to have those particular bacteria. Okay, one more story about initial colonization. So one of the really interesting things that I read was there was I want to say it was back in the 60s, maybe 70s, there was a strain of Streptococcus that was going around in some hospitals. And so babies were being born in the hospital, immediately picking up this bacterial infection, and it can be lethal, right? This was really bad. And so some researchers were studying this, and at one hospital, they found that it made a difference which nurse handled them. So basically, they found a nurse that was a carrier for the bad bacteria. And so if she was the one carrying for the babies, they were contracting this infection and having really poor outcomes. Super interesting. As long as she didn't handle them for the first 24 hours, they were fine. They were getting colonized by bacteria from other healthcare workers. And then they were strong enough that it didn't matter that they were exposed to this specific strain of strep. And so they were calling it gardening babies. They basically figured out what the good bacteria was and started taking it to variety hospitals and were able to wipe out this infection. Anyway, I thought that was fascinating. Okay, so initial colonization through the birth canal. Then really we consider all these subsequent colonizations. I mean, obviously you're in contact with healthcare professionals as you're coming out. Mom, of course, plays a big role here. And then also breast milk. They have found it's like 600 different species of bacteria that are actually in breast milk, which sounds crazy, but it's not, right? It's all these ducts and glands that bacteria can live in. And some of these are so important for the baby. So there's a genus, for example, called Bifidobacterium. that actually helps digest certain sugars and actually produces some acids that are really protective for the baby. So breastfeeding is super important. Not everyone can do it, right? Some babies are going to be bottle fed, but that does change colonization, right? Colonization, of course, from other family members, and from then on, it continues depending on what your environment is. Interesting studies. I think that first movie you watched, right, talked about kids and their contact with farm animals, right? And it's actually the microbes that they're in contact with and how that might make a difference in their immune systems functioning, and on and on. Okay. So, sorry, this is an old graph out of a different text, but I kind of liked it, right? So they're talking about this idea where we come in contact with bacteria, right? We were just looking at that initial colonization, but for the rest of your life, you're in contact. But here's the deal, right? Some of those bacteria are going to colonize us in a good way, right? They're going to become part of our microbiota, and we kind of have a pact, right, to work nicely with each other. Of course, there's a couple asterisks. Here's my pointer. Of course, there's a couple asterisks on that because occasionally, right, they can become opportunistic. So what we're going to look at, though, is sometimes you come in contact with those microbes, right, and they're foe right away, right? And so we're going to look here at what it looks like for a microbe to actually be able to invade and cause infection, right? What it looks like for a microbe to be a pathogen. Remember, a pathogen just means that we're looking at a microbe that's able to cause disease. Okay. When we talk about the pathogenicity of a microbe, we're talking about its potential to cause disease. So there's a difference between being an opportunistic pathogen and a true pathogen, right? Opportunistic pathogens, they are going to cause disease, say, when the host's immune system is weakened, right? It only happens when the host isn't doing great. A true pathogen is actually capable of causing disease even when the host is totally healthy, right? And so that's going to be a much more pathogenic microbe, true pathogen is. We'll also see that these bacteria differ, or I should say microbes, viruses, fungus, whatever, they differ in their what we call virulence. And virulence deals with the amount of damage that they cause on the host. And so we're going to look at a bunch of different factors in this chapter that might make microbes more pathogenic, might make microbes more virulent, and what this looks like as they're causing disease. So we had our moment for we love microbes, they're so nice, they're helping that little baby digest milk sugar, and now we're going to talk about disease. Okay, so figure 11.5, I come back to this one pretty often here, and this really just shows kind of the flow chart, right, of what the steps of infection are. I should have a little title here, Steps of Infection, right? And so we'll dig into these, but basically the microbe needs to find a portal of entry. It has to have a way into the host, right? You, your patient, whichever you want to think about it. Once it's in there, it needs to actually be able to attach to the site, survive the host defenses. It does then actually cause disease. It's not necessarily trying to cause harm to its host, but it is, right? And that's what the damage and the disease is. And then it also needs to find a way out of the host so that it can get into another host. And remember, it's not gonna have forever to do this because our immune system is going to come looking for it. So let's start by looking at this idea of there being a portal of entry, right? So what we're gonna find, this is not the slide I wanted at all. When we think about a portal of entry, you have to think about, okay, how are these microbes getting in? And so you might imagine like the skin is a really nice barrier, but if it's broken, right, you have a hangnail, you fall off your bike and, scrape some skin, that becomes a way microbes can get past what's normally a very solid defense. So skin, mucous membranes, some of them get in through the GI tract, right? So if you eat contaminated food or drink contaminated water, that is their portal of entry. That's how they've gotten in. Of course, right now we're dealing with a respiratory infection, right, where its portal of entry is actually through these aerosolized particles, which makes it pretty easy to spread around. Your urogenital tract, so urinary tract infections, sexually transmitted infections, those sorts of things. And then they're also pointing out this idea of endogenous bacteria. Those are the ones that you already had in you. So that would be an opportunistic infection. It's already there. But everybody else who's not in your normal microbiota is going to need to find a way in. Once they find their way in, right, they need to be able to attach firmly. And so this takes us back to some of the very beginning chapters where we talked about some of these appendages like fimbriae and capsules and how those could both help us stick to surfaces, stick to biofilms, that sort of thing. And then in the viral side of things, think back, it's actually the spikes. Remember spikes? They were surface proteins, kind of, particularly if you have an enveloped virus, they're going to stick out here, these little they look like little matches on here, right? Those are the spikes. They actually need to match up with a receptor on the host cell to get into that cell. So this attachment for a virus is super important. It's going to, right, we had to have the portal of entry into the body, say like a respiratory droplet gets into your lungs. But then if you have a virus in there, it needs to find the correct receptor in order to gain entry to the cell as well. So viruses have maybe an extra step in there, right? So portal of entry and then this attachment. And then, I did want to point this out. Notice that how many microbes you need to come in contact with in order to actually get a disease. And we call that the infectious dose. We've heard that term some with COVID. It varies dramatically. I mean, look at this, right? Measles, you only need a single viral particle in order to become infected. That's kind of crazy. Where cholera, you need, 100 million cells of bacteria. That's some gnarly water, right? That you're drinking there in order to contract cholera. They're small, so it's possible, right? Okay, so it varies dramatically. Okay, so our next step then is evading these host defenses, right? In order to become a pathogen, in order to actually cause disease, that microbe needs to attach to a surface and then to establish itself, to start replicating, it's going to need to be able to survive host defenses. And so we'll see a few different things. A couple of these I wanted to point out. Let me get myself out of your way here. This is a big one. So what we find in bacteria that can produce capsules, that capsule is a very good defense against being phagocytized, right? So this is say like a macrophage supposed to be engulfing foreign bacteria. If that bacteria has a capsule, phagocytes are much less able to take care of that. And so that's a real big one. Once you've evaded those immune system defenses, really we're at the point now where this microbe can cause disease. And so a couple of these are kind of pointing out some of these factors, right? The bacteria, in this case, are producing a variety of different enzymes to disrupt tissues. Now, this is part of the microbe's plan often, right? It's trying to get maybe deeper into a tissue. I clearly talk to myself in these recordings. So put myself back up here so I look up at you. These enzymes, right, are digging into deeper tissues so they can get to more of the resources that they want, right? So there's certain enzymes that destroy things like the connective tissue, right? So here we see bacteria getting between these cells, right? Epithelial cells always have connective tissue underlying them. And so we're trying to destroy that, right? You might have a bacteria that produces enzymes to destroy the mucus in a mucous membrane because that's going to let them get in deeper. And so that's a common technique used by bacteria. Bacteria are also going to produce a variety of different toxins. Here's right? They're just poisons. There might be neurotoxins, right, that damage neurons, hepatotoxins that damage the liver, nephrotoxins, those are ones that damage the kidney, enterotoxins would destroy the gut lining, hemotoxin, destroy blood cells, etc. One key thing to keep in mind, these toxins can really look two different ways. So the ones they're showing us here are called exotoxins. These are going to be toxins that are actually released from the bacteria. They tend to be really strong and have a very specific target. That would be things like a neurotoxin or a nephrotoxin, right? They're going after something specific. Gram-negative bacteria have embedded in their outer membrane a lipopolysaccharide, right? LPS. And this actually ends up being what's called an endotoxin. It's part of the bacteria. It's not trying to release it. It's just embedded in that outer membrane. And this is something that during gram-negative treatment, as those cells are dying, then this lipopolysaccharide becomes available to attack the host. And this can actually lead to fatal shock, right? So you have to be really careful with the gram-negatives. Right, so again, showing lots of different ways that bacteria are getting deeper. This one's crazy, right? So they're showing salmonella attaching here, and then it's actually interfering with the actin inside of the cell. So the cytoskeleton of this host cell, to put microvilli out, you actually use actin, part of the cytoskeleton. And so basically it's tricked the cell into actually bringing it inside in a vacuole. And that's actually how this salmonella, it just came into the microvilli, the small intestine, which is a common food poisoning. Notice then it's actually doing cell division inside of this cell, and then it's able to exit out the other side of the cell and really get into deeper tissues where it can cause a lot of problems, right? So as gnarly as all of these things are, right, I will point out that a lot of times, right, the disease, the signs and symptoms that we see, maybe a good time to remind you that there is a difference between signs and symptoms. Right? Signs are objective. Signs are things that a doctor can see or measure, right? Like you are running a fever, right? Put a thermometer in, 101. You have a fever. That's a sign. Symptoms are subjective. They're things that the patient reports, like, I feel like I'm going to puke, right? Or I'm nauseous. That is subjective, right? That is a symptom. When they actually vomit on you, then it's a sign, right? We know it was real. Not that symptoms aren't real, but they're not measurable, they're not observable. Okay. So as bad as some of these things here, different toxins are, I did want to point out that there's also a lot of indirect damage that takes place. And we'll talk more about this maybe when we get into the immune system. But basically, a lot of the damage is actually caused by the host having an excessive or inappropriate response to this pathogen, right? So as your immune system is trying to seek and destroy, sometimes it causes cell death, right? Your immune system is trying to kill bacteria and you kind of lose some normal cells as kind of like a friendly fire, right? And so that would lead to what we call necrosis, right? So cell death. Sometimes it's these bacterial toxins, but a lot of times it's the friendly fire. Inflammation can be a good thing or it can be really excessive and it can be bad. Same with things like edema. We'll see swollen lymph nodes. That's a really good sign of infection, right? So you could actually feel those. So that's a sign, and that tells us that our immune system is working. It's searching for these things. So I guess I shouldn't even preface this as trying to act like it's all the immune system's fault. Maybe I'll just say that a lot of damage is caused by an excessive or inappropriate response by the host. Okay. But as far as signs and symptoms go, we have things like inflammation, edema, swollen lymph nodes, leukocytosis, which is the fancy way to say an increased white blood cell count. Other things that we might see, bacteremia, that's bacteria in the blood. I don't hear that term near as often as septicemia, and that's when you have bacteria in large quantities in the blood, right? So if you hear about someone dying of sepsis, right, this is septicemia, bacteria, lots of bacteria in the blood. We can use the presence of antibodies as a sign, maybe that infection has passed, etc. Right? So there is a lot going on during that actual course of disease. So this graph, this is figure 11.7. Notice this looks an awful lot like the bacterial growth curve because they overlap, right? But notice what we're looking at now is time versus intensity of those symptoms that we were just talking about, right? So you have that initial exposure and an incubation period. And basically during this incubation period, that microbe is just ramping itself up, right? Remember the lag phase? It's not growing. It hasn't reached the power of exponential growth yet. So these do definitely overlap. But the incubation period, you're not going to know, right, that you're sick. And that's a tough one here in COVID because 2 to 14 days for an incubation period, that's a big range there and makes it kind of difficult to judge who's still going to get sick and that sort of thing. Okay. Your patient or yourself then moves into what we call the prodromal stage. And this is kind of that, it's generally described as general malaise. Like you're just not feeling good. You're kind of under the weather, you're tired, but no specific signs and symptoms, right? The period of invasion, now we're starting to see those signs and symptoms increase. And so again, some of this is what the bacteria or virus or other microbe is doing to you. And a lot of this is what your immune system is doing, right? Like we're going to get that virus. We just need to run a fever. You often actually turn the fever on yourself as a defense mechanism, right? So anyway, this is when you're actually sick. And then hopefully we enter what's called the convalescent period. Convalescence is the fancy way to say recovery. Right? Doesn't convalescent just sound cool? It's like Dr. Quinn medicine woman. I mean, people who had tuberculosis taking the train out west to hang out at the hot springs and convalesce. So there you go. It's a good word. Okay. Let's see where I am. Oh, signs and symptoms. I already talked about that. A few others for you there. And then keeping in mind that at the end of that whole process, this microbe also needs to have a portal of exit. It needs a way out through some secretion, excretion, discharge, sloughing, right? It's got to go somewhere. And this is because eventually your immune system, the host immune system, is going to catch on. It is going to rid the body of this microbe, and it's hoping that it's made it to another naive host by the time that you catch on. So, I thought I had. This is just some other random terminology that goes with infection, if it helps at all. Localized versus systemic, right? Localized, it's in one spot. Systemic is throughout the body. A focal infection, that's another good term. This is when you had a localized infection, say strep throat, and then it spread. So in this case, strep throat, those same bacteria can damage the valves in your heart. And so if you had like a myocarditis or an endocarditis, a heart infection, that would be considered a focal infection. It spread from elsewhere. Mixed infections, very common to have multiple different species of microbes. That can make antibiotic treatment difficult. And then primary versus secondary infection. So primary infection is going to be kind of this initial infection. You get the antibiotic for a primary infection, and that leads to a secondary infection. This is the one I was talking about earlier, where you end up with a yeast infection. So the easier way to talk about this is probably a secondary infection is secondary to something like antibiotic exposure. Primary infection is just a normal infection. Okay, sorry, a little tangent there about signs and symptoms. Okay, portal of exit. Isn't that lovely? Okay. Okay, so here comes a lot of terminology, but it's not terrible. Okay, So this whole table, so 11.5 here, is talking about reservoirs and transmitters. So a reservoir is where a pathogen persists, like where it can hang out and just be. So a lot of times we talk about kind of the primary habitat in the natural world where this pathogen originates, right? So people want to know, like, what is the reservoir for SARS-CoV-2, the virus that causes COVID-19? Like, it has to have come from somewhere. Where does it hang out? Okay. So these reservoirs, the places basically where these microbes can hang out, they can be animals. And they point out here other than humans and arthropods. So we'll give specific terminology here for humans, and we'll give some specific terminology here with arthropods, which are insects. So bats, and other mammals like, I don't know, rats spreading the plague, or prairie dogs on the Front Range, right, have the plague, birds, reptiles, pangolin, wherever that is, right? They're blaming that on for COVID-19. Humans can be reservoirs, right? Places where these microbes are hanging out. So someone who is actively ill, and this is a really tough one with COVID-19 because so many people are asymptomatic. They're sick, but they don't have the signs and symptoms to tell them that, right? But chewing on your pen and handing it to someone else to sign a paper, hey, that's how we used to spread the common cold. Maybe this will be a good thing. We don't do that anymore. A human who is recovered but might still be spreading, or I suppose someone who's asymptomatic, I would call them a carrier as well. So someone who's actively ill, showing signs and symptoms, I guess, versus someone who is a carrier. So they spread it without notice. So COVID-19 is a great example. People who are in the incubation period or the prodromal phase are often carriers of disease. And you think about some things like HIV, that could actually have a really long incubation period before people know, but they would still be able to transmit that disease. Sometimes people in that convalescent phase, they feel better, but maybe they're still shedding that microbe. And then there are people who become these chronic carriers. So have you guys heard of Typhoid Mary before? This is like, I want to say 1800s, maybe early 1900s. She was a cook. She carried typhoid fever. It never made her sick, but she kept working as a cook, and it spread through fecal oral contamination. And so people that she worked for kept getting ill. A few of them died. She would get arrested, moved around. She didn't believe it, kept spreading it. Anyway, so it's not good to be a Typhoid Mary. That means you're a carrier. Anyway, then other reservoirs here, basically thinking of these insects, these biological vectors. So how do I want to say this? So here's a mosquito, right? Really well known for transmitting things like malaria. It can carry that from person to person. And so it's very common for mosquitoes in the wild to have these diseases in their saliva, being able to spread them at will. Other biological vectors, I kind of don't love how they did this, right? So they split out insects, but really I tend to think of biological vectors, like including the insect, including the bat or the pangolin, right? Because what we'll find is that a lot of these new diseases are what we call zoonoses, or a zoonotic disease. They come from animals. So they end up spreading to humans, but they came from animals. They're saying right now about 70% of all the emerging infectious diseases in the world are actually coming from animals. And I will point out that insects are animals. So this is kind of an awkward way that they've split it up. But the arthropods are very important. Ticks, mosquitoes, there's some other bugs. Those are the main ones we have that will spread disease. Okay, non-living reservoirs. You can also have certain diseases or certain microbes that can hang out for a while in soil, water, air, the built environment, right? This was a big thing with COVID, like how long can it survive on cardboard? Do I really need to wipe my groceries down, right? Those sorts of things. Do we have non-living reservoirs? And some bacteria or some microbes, sorry, I keep saying bacteria. Some microbes are better than others at surviving in non-living reservoirs. Viruses are terrible, right? They need to be in a host cell in order to survive and reproduce, so they don't live that long outside. Here they're pointing out tuberculosis can actually hang out for a really long time. Okay, so this is a whole table on carriers. Look at this. So we mentioned carriers right here. Now we're going to blow this up. So next table 11.6. Again, when we're talking about carriers, this is an individual who carries without notice, but is able to spread the disease. So asymptomatic carrier, right? They don't have signs and symptoms, so they don't know. This is particularly this is their example, right? Asymptomatic STDs. That's very common. A lot of STIs don't even cause symptoms in the majority of people, or some of them have very long incubation periods, right? And so, yeah, absolutely. We have asymptomatic carriers. Here's someone who's incubating, right? They don't know that they're sick yet. Convalescent, this lady is getting better, right? But maybe she still has some hepatitis A that she'll be shedding for a day or two, right? Wash those hands. Chronic carrier, this is typhoid Mary. It's not, but they're pointing out typhoid fever and she's a cook. They're going for something here. Okay. This one's probably important to mention as well, since most of you are going into healthcare, this idea of passive carriers. And Jamie was kind of talking about this with that idea that a lot of medical professionals actually carry MRSA, right? And it's not making them sick, but they can hand it off to their patients. And even more than that, right? Like the need to wash hands. Here we see this healthcare professional emptying I think that's a bedpan, but clearly she still has the germs on her hands when she goes to treat this patient. And so, again, really good hygiene, glove wearing, hand washing, and alcohol hand gel, all those kind of things, right? Just realize you can pass these on. Okay. Wow. Okay. Vertical transmission versus horizontal transmission. So vertical transmission, we talk about when a disease is moving from a parent to the offspring, right? So for example, if you have syphilis as a female, it can cross the placenta and infect your baby. Your baby can be born with syphilis. That would be a vertical transmission of a disease, right? Or if you have HIV and you breastfeed your baby and they pick up HIV. Okay, horizontal transmission is where we're going to spend most of our time here. Again, just a ton of terminology. Check out these bold print words. That's primarily what you're looking for. Okay, so in horizontal transmission, it's basically direct, indirect, or vector-borne. So direct transmission requires actual physical contact, so shaking hands, having sex, you know. They do include here droplets. So not aerosols, right, where it might be like hanging out in the air for a really long time, but like this lady just straight up sneezed in her face, and that's gross. Okay, so direct contact. Indirect transmission is a little bit more difficult, right? So notice a few more key terms. Fomite. So a fomite is an inanimate object that harbors and transmits the pathogen. The doorknob is the perfect example, right? Someone, rubbed their nose, they went out the door, the next person who opens that door, then, scratches their eye, they can transmit. So a fomite. Look at so many good words. Convalescent fomite. Vehicle. This is going to be an inanimate object, right? A non-living object that can transmit. Really important vehicle, water, all of these. So air, here they're including aerosols. So this is kind of the COVID-19 thing. This might be able to hang out we're still not positive, but it's probably able to hang out in kind of a fine suspension for a long time. So air is acting as the vehicle of transmission, right? You could walk into a room 10 minutes after someone and potentially still pick up that disease. Water, this is a huge one. Cholera, a ton of different waterborne diseases. We'll talk about some. Soil. Soils can act as vehicles of transmission. The only one coming to mind right now are prions. Prions seem to be able to survive in soil. So they had an elk farm down in like the Fort Collins area that had a chronic wasting disease outbreak, and so they like culled the herd. They killed them all, moved them out, and then they brought a new set of elk in, and they all got it, and that's when we realized it spread in the soil. Food, right? So again, in the preparation of food, can also be a good way to pick up stuff. And they point out especially oral fecal route. So this is a big thing in food preparation, is that you gotta wash your hands after you use the bathroom, right? So there's plenty of these diseases. Their portal of exit is through feces, and if you don't wash your hands, I mean, you don't have to have visible poop on your hands. All you need is a little bit of bacteria. It's microscopic, and then you can get that into someone else's food. Okay, vector transmission. Again, biological vectors, insects are the perfect example. A mechanical vector, insect carries the microbes to a host on its body parts. Oh, so that's different than a biological vector? So biological vector, they're saying you actually have to inject it. So like a tick or a mosquito biting you, as opposed to if now I have to look. I think when fleas carry the plague, I don't think they have to bite you to pass on the bacteria. I think the plague bacteria is just on the flea, and then that can be delivered to you. I would still consider it a biological vector, but I'm not in charge. Okay. Sorry, just pointing out, just making sure, right? It's really important, particularly right now with COVID, to recognize the difference between droplets and aerosols. Okay, we can talk in class if there's more questions on that. Okay, so then one of the things we should really consider, particularly because we're going into healthcare, is this idea that the hospital or the healthcare setting can be a source of disease. And so there's a special name given to infectious diseases that are acquired during a hospital stay. And the term there is nosocomial, Nosocomial, nosocomial infections. And that's what this graph is showing. So we have something like 2 to 4 million nosocomial infections every year, and about 100,000 deaths due to infections that were picked up at the hospital. And so this graph is showing where those fall out. Those of you already working in healthcare, you know urinary tract infections, particularly in individuals who have catheters. That's going to be a huge portion of this, right? But again, we see surgical sites getting infected. We see pneumonia and other respiratory infections. There's the septicemia, right? Getting blood, bacteria into the bloodstream. So this is a big problem, right? Our patients are compromised, so they are much more likely to pick up infections. They're likely to get opportunistic infections. There's the reuse of equipment, so we do need to sterilize things properly. And like we were talking about last week, antibiotic-resistant strains are really common. And so that's, it's one thing to get a urinary tract infection, and it's another to get a UTI that's multi-drug resistant, right? It's one thing to fall on your mountain bike and get some dirt in a scrape, and it's another to get MRSA in a surgical site. So again, being really careful about asepsis, about keeping things clean, about changing gloves. I've heard horror stories. And we wear gloves now, right? This was actually something really good, probably that came out of the AIDS epidemic, is we moved to this idea of universal precautions, right? We treat everyone as if they are infected with HIV, which means we're wearing gloves, trying to limit the spread of bodily fluids. And now we're in a whole nother realm, right? This thought about, what does the asepsis look like, or what do the universal precautions look like when we have an illness like COVID-19? And I think we have yet to see how that all pans out. Okay, and the final portion of this chapter deals with epidemiology. We've heard about this a lot in the news. Epidemiology is the study of disease in populations. So they're looking at things, an epidemiologist is looking at things like the frequency and the distribution of disease. And they're looking for things, right, that are out of the ordinary. Now, the two main epidemiological groups, I would say, are the CDC, the Centers for Disease Control and Prevention, and that's based in Atlanta. That is a U.S. government organization. They used to would normally work very closely with the WHO, the World Health Organization. I don't think we can call them the WHO because that's a band from the 70s. So let's keep it WHO, World Health Organization. They work really closely together, but the CDC for sure has an amazing set of resources, and they're often called on. Right, like we send crews over to Africa when there's Ebola outbreaks and try to understand the spread of cholera in other countries. So the CDC is pretty amazing. So this is just an idea of an example of some different kind of epidemiological reports that you might see. Oh, I should totally show you, remind me, I'll show you Bug Watch. It's a really good one here in Colorado. A couple things they're trying to show you here, right? So we're graphing the distribution of disease, right? And so, and I'm clearly missing something in the footnotes on figure 11:10 as to why these graphs are all different. The things we tend to be looking at are things like prevalence. That is the percentage of the population with that are cases, right? So a percentage. Incidence, which is the number of new cases in the population. Morbidity is the number of people who are ill. Mortality is the number of people who have died, right? So again, a bunch of terminology. Prevalence is a percentage. Incidence tends to be like out of 10,000, right? It's according to the population. Morbidity is illness. Mortality is deaths. So the CDC actually puts out a document called Morbidity and Mortality Weekly, and I can show you an example of that in class as well. Okay, last few terms here. Endemic. So there are some diseases that we would say are endemic. So there's like a place, a location where the disease is present over the long term. Often this is due to the vector, right? So we'll talk about, say, like areas of South America where malaria is endemic. It's just been there, right? And so you may have a lot of cases, but that's not surprising to an epidemiologist, right? That's just like, those are the normal numbers. What we're looking for often are things like an epidemic. Now, technically an epidemic is just the rate of disease beyond what we expected, okay? So SARS-CoV-2 is an epidemic as soon as it starts because we were expecting 0 cases. We did not know about this disease, right? But when you hear about outbreaks of, say, Ebola in Sierra Leone or something like that, that's an epidemic. It's beyond kind of those background rates of infection. If something has sporadic occurrence, it's just popping up occasionally. We don't necessarily know why. Pandemic, of course, is an epidemic that's spread over multiple continents. We all know way too much about pandemics right now, right? So again, the epidemiology can be super useful in tracking these disease outbreaks. So here we're looking at foodborne illness, right? Salmonella. We're starting to get reports. There are certain diseases that healthcare hospitals and doctors need to report, right? They're considered reportable diseases. They have to send this information to the CDC or to public health so that it can be mapped, right? So that we notice that something different is going on. Like, even this is interesting. So incidence, again, we're looking at like per 10,000 people. And what we're seeing here is like what's going on in 2008, right? So they've zoomed in and they're showing us here what's going on in 2008. And then you can start, right, when you realize you have this spike, then you can start trying to figure out, okay, if you know something about salmonella and how quickly it replicates, like this is probably not right? When people are getting salmonella, but we might need to look back at like, was there a state fair and a vendor there? Like what happened? Okay, and so that's where this stuff is super nerdy, mathy, graphy, but it's super important. And you will see that countries that have done well with COVID-19 are doing a really good job with epidemiology. And we have the CDC, and we're not. Okay, so the CDC puts out a ton of really great information. We can walk around their website a little bit in class, but this might be super useful for you in the future on any reports and things that you need to do. Maybe you're in nursing school and you have an assignment about AIDS. Great visuals, great graphs, huge amounts of data. Okay, that's it. We will see you on Thursday.